In this video, we're going to look at the factors that influence protein stability, including hydrophobic effects, and eventually we're going to look at denaturation and unfolded regions. So typically a 100 residue protein isn't all that stable in its native conformation. We say it roughly has a delta G of 40 kilojoules per mole. To put that in perspective, what that means, a hydrogen bond has roughly 20 kilojoules per mole of stability. So a folded protein is twice as stable as a hydrogen bond. So it's not all that stable. Looking at the various interactions that stabilize the native conformation versus the denatured conformation, we have hydrophobic effects. We do have electrostatic effects, not as important, but it's important when they're inside of a protein, or what I call buried. And hydrogen bond effects, again, this is very minor, and we'll see why that is in a second. So the main reasons why proteins fold and why they fold the way they do is because of the hydrophobic effect. Now, we have nonpolar amino acids, and if you remember, if a nonpolar substance is in contact with water, water will form these cages around the hydrophobic substances. They don't want to interact with hydrophobic molecules because they cannot hydrogen bond with them. So what happens in a protein is that all these hydrophobic residues are going to be pushed towards the center, and they're all going to be what we call buried, or in the interior of a protein, while the polar and charged residues will be on the exterior because they can hydrogen bond with water. And we can rank the likelihood of an amino acid being in the interior, and that's called hydropathy. Now, and we have an index for that, hydropathic index. And so how to read this chart is the numbers, the positive numbers are amino acids that are likely to be buried. Negative numbers are residues that will not be in the interior, and it makes sense, right? Isoleucine, valine, high hydropathy numbers, lysine, arginine, negative numbers. And you can actually create a scale of this hydropathic index where you look at the residue numbers, and it's kind of a running average, where you just look at, okay, what type of amino acids do we have here? So if you get high positive numbers, that means these amino acids are likely buried. If you get negative numbers, that means that these amino acids are likely to be on the exterior. And when you think about the core of a protein, it's not just hydrophobics that dictate what's going to be in the core or how the core is arranged. You also have to think of steric hindrance and the volume of these side chains, and that really dictates how they'll be in the core, and that'll dictate how a protein folds. Now, aside from hydrophobic effects, we also have ion pairs or electrostatic interactions and hydrogen bonds as well. So hydrogen bonds, be it through the backbone or polar molecules, overall they don't really play that big of a role in stability. And that's because hydrogen bonds in proteins, they can hydrogen bond with the water or they can hydrogen bond with amino acids. So it really doesn't matter if you're folded or unfolded to do that. But they can help adjust the structure. So when a protein's folding, and all the hydrophobic residues are coming together, the structure might have to adjust a little bit to form hydrogen bonds. That's what I mean by fine-tuning. Ion pairs and salt bridges might be important as well. So if you have a negative and a positive amino acid in contact with each other like this, we call that a salt bridge. Now, a lot of ions are actually found on the outside of a protein, but it is possible to have salt bridges on the interior of a protein. It's very important that these amino acids are paired because an unpaired charged amino acid is very destabilizing for a protein. And there is some penalties for having ion pairs. So if you have an ion pair, you're going to get a loss of entropy because these two amino acids have to be next to each other. You can't get any rotation around your carbon bonds anymore. You're going to lose some solvation penalties as well. So normally these ions would have water all around them. That no longer happens when you have a salt bridge like this. Ion pairs, though, if you have a multi-subunit protein, like we'll see for hemoglobin coming up, these ion pairs can be very important so the different subunits can talk to each other. Talk is in quotes, so these subunits can interact with each other. Now, another important type of bond for structure is disulfide bonds. And these will form as proteins fold. Two cysteines can come together and you can form a disulfide bond. Now, there has been some mutation studies to try and see, are cysteines, are disulfide bonds critical for the actual 3D structure of a protein? And what you see is that they might not be essential for activity. That is, we can mutate the cysteine to something else, and the protein will still be active. It will still do its function. So, for example, if we take cysteine to serine, the protein still works. The reason why disulfide bonds can be important is that they will lock in certain conformations. And really, disulfide bonds you will not see in intracellular proteins. That's because the cytoplasm is what we call a reducing environment. That is, any disulfide bond that forms, they will break in the cytoplasm. However, outside the cytoplasm, that's where you really see these disulfide bonds, and that actually makes a lot of sense when you take a second to think about it. Inside the cell, it's controlled. You have a buffer, you have chaperones, which we'll talk in a little bit. Basically, this environment is ideal for a protein. Outside the cell, anything can happen. You can get swings of temperature, swings of pH. So this disulfide could be an extra layer of like glue to try and hold that protein together. So that's why it might be more critical on the outside proteins, the extracellular proteins. And when thinking of forces that hold proteins together, we actually see some motifs that come out of that. And one is through zinc fingers. And what a zinc finger is that you have a metal ion. Here we have zinc, and that actually attracts different amino acids to this metal. Now, for zinc fingers, it's always going to be zinc, but we see behavior like this with different types of metals. So here, zinc can coordinate to cysteines, histidines, aspartic acids, and glutamic acids. Usually, it's going to be in cysteine and histidines. That's what we're seeing here: 2 histidines, 2 cystidines. And this zinc, the way that these amino acids interact with zinc, it actually favors this type of folding pattern. So the zinc is important for 3D structure. And if you take the zinc away, this protein will actually unfold. And you'll see the zinc finger motif in proteins that bind nucleotides. This motif is critical for a protein's ability to bind to like DNA, RNA, or nucleotides in general. Alright, that's it for this video. I will see you in the next one.